Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Today's conversation is one I really enjoyed because it reminds us why many of us fell in love with science in the first place. My guest is Keyvan Sedaghat, a pharmacologist, educator, and a lifelong GPCR thinker whose journey started with childhood experiments and grew into decades of teaching, research, and reflection. In this episode, you'll hear how curiosity, not hype, drives lasting scientific impact. We talk about GPCR regulation, desensitization, lipid rafts, and why the so-called classical paradigms keep breaking in interesting ways. You'll also hear why teaching sharpens scientific thinking, how focus shapes a career, and why GPCRs still have so much left to reveal. The Dr. GPCR University works best when it's shared. The university brings the global GPCR community together through expert-led courses and open contribution, all within the nonprofit of the Dr. GPCR ecosystem. Every voice helps shape what comes next. Join in by clicking on the link in the show notes. If you ever wanted Dr. Terry Kinakin as a mentor, this is the next best thing. Terry's Corner delivers weekly, bite-sized lessons, monthly Ask Me Anything sessions, and frameworks that actually move the needle in discovery. Built for scientists who think the long game. Start with a trailer. Links are also in the show. This episode and the Dr. GPCR ecosystem is supported by our collaborators. If you're running GPCR binding or lifestyle assays, Celtaris Research offers fluorescent ligands designed to replace radioactivity while preserving kinetic insights. For sharp, reproducible GPCR assays, check out Celtaris Research's link, also in the show notes. And today, I'm especially excited to welcome Eurofins DiscoverX as a new strategic partner for the Dr. GPCR ecosystem. If you're developing GPCR therapeutics, assay choice isn't a detail. It's a decision that carries through the clinic. Eurofins DiscoverX delivers industry-standard GPCR assays trusted for hit ranking, bias analysis, and regulatory submissions as well, built to scale with real drug discovery programs. All the links are in the show notes. And now, let's dive in.
Hello, everyone. This is Yamina from Dr. GPCR, and today I'm very excited. Finally, we got to do this. I'm excited to have with me Dr. Keyvan Sedaghat. Welcome to the podcast, Keyvan.
Thank you so much, Yamina. It's a great pleasure to be here. Thanks for the kind invitation. It's been really an honor to be here because I know big names have been on this podcast. So I know it's the kind of awkward position to talk about science because of these big names, but I do my best.
Thank you so much.
You are welcome. I wanted to thank you for this great job you are doing and your team because it's very important and valuable for dissemination of knowledge because the generation of knowledge, application, and dissemination is an important, I think, part of when we deal with knowledge. And I really appreciate your hard work.
Thank you so much. We really appreciate it. We do this day in, day out because we love what we do. And hearing such kind words always motivates us for sure to continue.
Great.
All right, so let's start at the beginning. Could you please introduce yourself, tell us who you are and what is it that you do?
Okay, sure. Actually, I did my undergrad degree in Iran in pharmacy, and then I moved to Canada to pursue my graduate studies. So I did my master's and PhD at the University of Ottawa, Department of Cellular and Molecular Medicine. And that's where I became basically acquainted with G-protein coupled receptors because I did my, the thesis under supervision of Professor Mario Tiberi. So he was my advisor, and I was really privileged to get to know him and learn from him. And he was not my supervisor. He was also a great friend for me. We still are in touch, and we have a great friendship. We built a great friendship.
And Mario is wonderful. He actually was one of our co-organizers for the Great Lakes GPCR retreat actually 18 months ago. So I got the chance to work with Mario and the Ottawa team to organize the meeting. So that's wonderful.
Yeah. So I think to be honest with you, I don't want to speak cliche, but for my passion for science started from early childhood while I was in elementary school. So I was always interested in knowing, about nature, sky, stars, and plants, animals. And I remember I made a small water electrolysis apparatus with simple things while I was in the elementary school, and later I made a small electroscope to detect electric charge with a penicillin vial. and a few pieces of aluminum foil. So I was always interested in doing science and doing experiments. I had in mind wearing lab coats, working, in lab for long hours and making discoveries, so like anybody else. And also, I was very inspired about the life of scientists like Louis Pasteur or Santiago Ramon y Cajal, great scientists, and reading their stories. I had a book, I remember, that my mom bought me, that it was on the biography of different scientists. I really liked it, and it really inspired me to become a scientist.
I love it. And was there anybody else in your family that was a scientist, or it just came through knowledge, through learning?
Not, well, I have also cousin, but not my brothers and sisters, not anyone, very close, I would say. So
Interesting. And
I'm the only one who got interested in science.
interesting. So you mentioned really, reading about Louis Pasteur and these discoveries. But when you were a child, did you think you'd be like a lab scientist, or was there, sometimes our guests want to be astrophysicists or astronauts. Was there anything specific you had in mind, or it was mainly just asking questions about nature and understanding your environment?
Yeah, to be honest, I think more a lab scientist doing experiments. Maybe I was more interested in chemistry, mixing compounds together and generating magic compounds. Yeah, that's what I was interested more.
Interesting. And then, so you moved to Ottawa. How did you go about, figuring out what kind of lab you wanted to move to, especially that you did 2 degrees at the University of Ottawa? Was there anything specific that drew you to the labs that you worked in, or it was either the team or the PI or the university itself?
Yeah, to be honest with you, because I did my undergrad in pharmacy, so I was looking something related to pharmacy. So at the Department of Cellular Molecular Medicine, they had a pharmacology program. So that program was somehow related to pharmacy, especially molecular pharmacology. And I was very interested in, knowing how drugs work in our body at the cellular molecular level. Because I remember when I was studying pharmacy, we didn't know in depth about the mechanism of action of different drugs. So, for example, in pharmacology books, you could see that the mechanism of action is not known, or they think it's going like doing vasodilation or a very vague mechanism. We didn't know what's going on at cellular and molecular level at that time. So I decided to go to a pharmacology program. I think that was maybe the most relevant program that I could find at that time.
Wonderful. And after you completed your degree, what was next for you? Did you think about, should I go into academia or industry, or was there anything specific that, that's the fork in the road for some?
Yeah, I'm always very passionate about teaching. After I got my high school diploma, I was teaching like private tutoring to make my living. I was very passionate. So I started teaching after getting my degree, and also doing a little bit of research as well.
Wonderful. I also actually was teaching in high school. I really always enjoyed teaching. So my sister was my first, let's say, victim when I held her in elementary school. But she had a friend, and her parents needed some help with their daughter. And so actually that's how, and I think at the time I was paid, I think $20 an hour or something like that. When you're a high school student and you get $20 an hour. But to be honest, it wasn't about being paid that amount. It was more about knowing that that little girl was able to go into the school she wanted to go, and she succeeded. And that feeling of seeing others, succeed that's the priceless part of it.
Yeah, I think teaching is really great because I think it's a learning process itself. You have to prepare for the lectures. You have to know the topic in depth, and also I think every day I learn, because I'm still teaching. I'm a lecturer in pharmacology in a private institute in Toronto. So I learn every day from the trainees and students. I think that's a great opportunity when we teach.
That's wonderful. So when you got to Canada to doing your degree in the lab with Mario, how did you go about picking out your project? What did you find very interesting? What kind of projects were there available in the lab, and how did you end up working on the project you'd actually worked on?
Yeah, truthfully, when I got into Dr. Tiberi's lab, there were two aspects of GPCR signaling that were, under study. One was a structure-activity relationship studies that we swapped different domains of, different GPCRs. At that time, we were working on dopamine receptors. And also another side was GPCR regulation, like trafficking and phosphorylation, desensitization. So, the project that I was assigned to was related to GPCR regulation, especially D1 receptor pharmacology. Because D1 is a substrate for different GRK isoforms. It's been shown that can undergo phosphorylation by GRK2, 3, 4, 5. So yeah, I was focusing on the desensitization of D1 in HEK293 cells with GRK2 and 3. And GRK2, 3 are closely related. As we know, sometimes people think they do the same job. But yeah. So what I found was that they are really different. And you see a different profile in the desensitization of D1 receptor. So at that time, we used like a serial deletion approach. We deleted domains of the cytoplasmic tail of the receptor, and we were looking at the different residues that are phosphorylated and how the receptor behaves when the cytoplasmic tail is missing. I think we found interesting findings there, because the receptor, when it's phosphorylation deficient, no phosphorylation, is still desensitized even better than the wild type. So I think we still think we know everything about GPCR, even the basic stuff, even the conventional paradigm, the classical paradigm. But as you know, it's been cracking for many years. There are exceptions in every step of the GPCR signaling. And I think that's the most important thing and interesting thing, that we learn everything about these receptors. So we are not, at a state that we say, okay, we know everything and we have targeted everything, and we should, go to other targets, maybe use other molecules. But really, it's not the case. And I think they have a good potential for, discovering new drugs.
No, I agree. And I do remember these studies where in the beginning people were deleting different portions of the receptor, parts of the C terminus, or doing alanine scanning, or parts of the N terminus, especially with, some of these receptors have very long N-terminal tails. So deleting them and trying to make a sense of what are the domains that are important in desensitization for the C-terminal part. Absolutely. Yeah. So if you had to pick a favorite GPCR, would it be D1? Or is there anything?
To be honest, it's very important for me, because I did a lot of research on that. But to be honest, all of them are the same for me. I really don't want to pick one of them, because I think all of them could be exploited as long as we can use them for developing therapy and alleviate patients pains. So I think they are all interesting. But obviously D1 is very important for me, because I did all my PhD studies on that receptor. But generally speaking, I think all of them are important, and if we can exploit them for any kind of disease, that would be great.
Absolutely. So after your PhD position, what was the next step in your career? You mentioned you're currently in Toronto teaching at a private institute. How did you go from a PhD to now teaching? And clearly I can tell that you have a passion for teaching.
Yeah, after my PhD, I actually got a position at the Baha'i Institute for Higher Education. So I continued research, and also I started teaching, in pharmacy program. And that's where I got the chance to do a little bit of research on GPCR. So one of the main, I would say, achievements was that we generated a database for G protein coupled receptors and lipid rafts. So it's called 7TMR-TMR. So 7TMR-Raft.org.
I'm typing it live. This is the first time I'm doing this during a full podcast, but I will actually put this onto our website under our episode so that people can,
it's free, available to all the scientists. That's what's the goal, to collect all the information one of my students worked on that. She did a great job, collected all the information, summarized that, and put all the information in that database. I hope that people find it useful, because I think compartmentalization of signaling in GPCR is very important. And probably you are familiar with the work of Dr. Mark Rasnick, when he showed that G-alpha subunit is compartmentalized to a lipid raft. So it's basically segregated from the signaling partners, and that's one of the mechanisms proposed for the depression. And antidepressive medication seems to have an effect on G-alpha subunit and bring it out of the lipid raft, and so they restore the signaling by beta-adrenergic receptor. And I think they have developed a test on lymphocytes to monitor the action of antidepressants based on the fractionation of alpha subunit, how many percentage of the subunit is in the lipid raft or in non-lipid raft compartment. So I mean, these are not just concepts. They have been people have been trying to bring it to practice and use it for monitoring drug action and also for diagnosis.
No, and I think there's a theme here. So I just recorded last week a podcast episode with Dr. Michelle Halls from the university, actually from Australia, from Monash, and we were just talking about how she is fascinated by the positioning, or how GPCRs end up in different compartments, and how, for example, lipid composition or access to accessory proteins changes GPCR signaling. And today, actually earlier, I met someone also working on, not necessarily only on GPCRs, but also on the interaction between proteins and lipids. And we ended up obviously talking about GPCRs, and we were just exactly talking about lipid rafts and lipid composition, and how does that change. For example, it could change the lipid composition between disease tissue or disease cell versus normal cell can change, and that could affect GPCR signaling as well. So it's absolutely fascinating.
Yeah, to be honest, I remember that in one of the GPCR retreats back in, I can't recall exactly, maybe it was 2007 or 8. And actually there was a talk by Dr. Kuzumi, Akihiro Kuzumi, and he introduced his great project on the organization of cell membrane. And talk about, I remember he actually distributed like a DVD in GPCR retreat, and I still have it somewhere. And it has,
you know what? I think I was at the retreat.
There's a great animation and about all his, the organization of lipid rafts, how the cytoskeleton works, and the interplay between the cytoskeleton and the microdomains of cell membrane. It was very fascinating for me, and that was I got interested in lipid rafts, cavioli, and I pursued that. And I also, for my comprehensive exam, for PhD comprehensive exam, the topic of my, grant proposal was also D1 signaling and lipid rafts.
Wonderful. And I think I was at that GPCR retreat. I think this was in London, Ontario, right? Maybe.
Probably.
And 2007, 2008. I do remember getting a CD from someone as well, or a DVD, and looking at it. At least I've been at not all, but a lot of GPCR retreats. So
it's very cool. I think that I still have the DVD at home, but it has movies on single molecule imaging, how this molecule, they hop different domains and they hop the cytoskeleton. It's very cool. I think the way they explain it, and very high tech research they are doing. I think it's a great book.
No, absolutely. And I think so. This is what I was talking with the other person today about the meeting they're organizing. By the time this podcast episode airs, the meeting will already be done. But what I was just telling the person is that unfortunately for a long time, we didn't have the tools. And now people are more and more interested in building out those tools and capabilities to look at receptor conformation and the receptor conformation landscape in the context of different lipid compositions. Unfortunately, you can't look at it in a whole cell, at least not yet.
Yes.
But I think it's really fascinating, and it's important to understand how does lipid composition change receptor function.
Yeah, to be honest, I did some research. We tried to actually deplete cholesterol in some animal models and see if there is any change in signaling. Well, we were looking basically at like a swing force test to see if the mice, which is depleted in cholesterol, will show some symptoms of depression. But it really didn't work, to be honest with you. I think there are very robust mechanisms in place that doesn't allow the composition of lipid composition of the neurons to be changed. It's very hard to manipulate the level of lipids in the membrane, unless you take like a genetic approach. You knock out a gene or something like that. But we were just using pharmacological tools, using like atorvastatin and statins to bring down the cholesterol with a very high dose, but it didn't work. That was the goal, to change the composition of membrane and see if there is any effect on the activity of receptors and we see in clinical signs.
It's a it's a tough thing to do, and especially with native cell lines like neurons. I mean, they're so sensitive that they're not amenable to these. So the person I was talking to, they're using this micropolymer that allows you to extract the receptor. You no need for detergent, no need for anything. It just extracts the receptor from the membrane with the the lipids surrounding it. I don't know exactly how it works. I learned about it about an hour ago. I haven't looked into it, but I thought it was a very interesting way of thinking about it and having these biophysical methods to look at the conformational changes in the receptor in the presence of a variety of lipids, but also in the presence of a variety of different concentrations, for example, of ligand.
I think these days people also are using molecular modeling and molecular dynamic simulation. So you can basically change, virtually the composition of membrane, the number of, for example, cholesterol or other lipids in the membrane, and see if it has any effect on the behavior or the three-dimensional shape of the receptor. I think this is something that could be used, but obviously doing the work in the lab, wet lab, is always something that should follow the virtual
computational studies. Yes.
Yeah, exactly.
Absolutely. I think sometimes we're way ahead of our time on the computational aspects, and we need a little bit more time to develop the methodologies to test out the theories that we have.
Well, I think extrapolating from computational studies to like wet lab or to real situation, we have to be very careful. Although there are a lot of tools developing these days. For example, we know that with AlphaFold, the prediction of the three-dimensional structure of proteins, because especially for GPCRs, it's very hard to, resolve the crystal structure. We know that only a few of them have been resolved. We have a big gap between the rhodopsin and also the next one that was beta-2-adrenergic receptor. So I think this AlphaFold, actually they got the Nobel Prize in Chemistry in 2024 for this computational protein design, and also for prediction of 3D structure of proteins. And also I think in physics, the two scientists got the award for using neuronal network and AI. So all of this together, they are all new tools that we have to use, makes things easier, but at the end of the day, you have to go in the lab and test everything and, see if they are real or not.
I agree. Again, you're absolutely right. It's all about, computational methods are, I think, fantastic. They're instrumental because they allow you to very quickly test a lot of scenarios, but to narrow it down and then to see what happens in reality, you have to run the assay. Develop the assay and run the assay to test the hypothesis for sure. Absolutely. So after you went back to Iran and you were teaching, you're back now in Toronto teaching again. How did this opportunity come up?
Actually, I was collaborating with a friend who has a company as a institution. They are training the pharmacists, the international pharmacists that they want to be licensed in Canada. So they have to go through different steps of the Pharmacy Examining Board of Canada. So that's why I teach, it's called evaluating exam. So I'm a lecturer in pharmacology and biomedical science right now. But I'm also affiliated to the university that I mentioned. So I'm still teach online and also do a little bit of research. So one of the outcome of my research was this database that I mentioned. So if anyone is interested knowing about GPCRs and lipid rafts and cardiology, so they can go to this website and all the information regarding, they can browse the receptors, search the receptors. So they have been, organized alphabetically, and about the organism, about the methods, the mechanism of association of receptor to the Raft, what is the functional implication, everything is summarized there. And we hope we could keep it updated. So we are still working on that. So right now I'm teaching and also doing a little bit of research. Another project that we are working also at the same time is on bitter taste receptors. Mm-hmm. So bitter taste receptors, they have extra oral functions. So they are expressed in other tissues such as brain, gut, lung, and vessels. So they could be used for treatment of cancer, like asthma, cardiovascular disease. I think that's another interesting, I would say, point about GPCRs, that gustatory receptors for sensing bitter taste, you can use it for like cancer. Nobody could think of that. And right now we are trying to manipulate the natural ligand. We were looking at some databases of bitter substances to change their structure and use some molecular docking studies to come up with better ligands with a better affinity to be able to use them as ligands for studying bitter taste receptors, or maybe they have a potential for drug discovery. That's another path we are pursuing right now.
I think that's wonderful. And, from a bitter taste receptor perspective, I did record a while back, actually, a podcast with Professor Mashanif. And I really loved learning about taste receptors and getting a sense, because when you think about it, when you say taste receptors, you think about taste. But as to your point, they do other things. They're expressed in other areas of the body. And the more we can learn about these receptors, the better. And obviously, they have clinical applications as well.
Yeah, something that I found interesting, if you activate them, they induce apoptosis. So they could be exploited for treating cancer, for example. And I remember in Iran, because we are very strong in traditional herbal medicine, some people, old physicians in the past were saying, if you, for example, take something with bitter taste, for example, some kernels of some fruits, then they might be useful for your health. They didn't say why. So maybe, I don't know, that's the reason. Because if you take these things, you ingest them, maybe they have like a preventive role. Yeah. I don't know, prevent cancer or something like that. Maybe that was the main reason. But they have many more potentials for other diseases as well.
Absolutely. And what I find fascinating about these, let's say, grandma type remedies is that people back in the day tested things out. Things worked, but there was never really a, deep research into the molecular mechanism. They just knew it worked.
Yeah.
Turmeric, for example, or garlic.
Exactly. Yeah. So. The apricot, you have in the center the kernel. It's very bitter. So some people say if you take 3 or 4 of them, they are toxic, but very, very little amount of it, maybe it's useful for your health. Maybe that's the reason. They activate the bitter taste receptors.
maybe somebody should be working on that and testing that out for sure. Wonderful. What were some of the top 3 aha moments that you had as a scientist that, kind of guided your career up until today?
Yeah. Truthfully, for me, when Dr. Lefkowitz got the Nobel Prize in 2012 in chemistry, that was I think that for me was a very important moment as a GPCR scientist, because I think our field was recognized, and it showed that it's an important field. And he, as
Which we
the
already
godfather
knew,
of
by the way.
yeah.
So the world knew as well. Apologies. I didn't want to interrupt, but I needed to make that comment. We knew, now the world knew as well.
Yes, exactly. I think they should have, awarded this the prize maybe sooner than that. But I think the most important thing is that the field of GPCR has been recognized by this award. Well, as you said, more than 30-40% of medications on the market are targeted, against the G protein coupled receptor. So it shows that they are very important. I think that's one of the moments that was important for me. But personally, knowing my advisor during PhD, Dr. Mario Tiberi, was a privilege because he really shaped my character as a scientist. I learned many things from him. And the way he was thinking, or the way I'm thinking today as a scientist, I learned everything from him. So that was also a great moment for me. Obviously, finishing my PhD and also the website that I talked about, these are great moments for me, just personal achievements, I would say.
Oh, absolutely. Thank you for sharing that. And we will share this website. And if you don't mind, I also will make an introduction to the person I was talking to who's interested in GPCRs and lipids and GPCR conformational changes. I think you're going to have a lot of commonalities there, and maybe they can benefit from the website, but also, who knows, maybe a collaboration might come out of it.
Hopefully. I hope they find it.
Yeah, absolutely. If you had advice for junior scientists who want to contribute to the field, what would that look like for you?
Well, because I teach a lot and I deal a lot with, newer generation, the first thing is that I want to advise them to do what they really love. I just wanted to mention the quote by, I think, the American physicist, Richard Feynman, when he was asked why he put so much effort and time and a big portion of his life on quantum physics, and why. And he said that because for the pleasure of finding out. I think we have to do research and science for the pleasure of finding out and solve the mysteries, I think. That's my first advice. Also, focus is very important. Everybody these days is looking for a formula for success. I remember I was reading in a paper about Dr. Lefkowitz. He was interviewed and he said there are 4 keys to success. The first one is focus. The second one is focus. The third one is focus. And you got to figure out the fourth one. So focus, I think, is very important because these days there are a lot of distractions for the younger generation. So that's why I think focus is very important. And from my personal experience, hard work, discipline, respect, and patience are the important factors for, I think, success in science. These are things that maybe newer generation, they don't like it very much compared to my generation. So they want to do things very fast. They are not patient. And, respect is very important for their peers, for their, teachers, advisors, everybody. These are, I think, important things. Also, I think they have to take advantage of any opportunity for learning and keep keep themselves in learning mode. But again, I remember the quote by Dr. Anthony Fauci. He said, "I'm a perpetual student." And I think we have to be a student perpetually and keep learning every day.
I agree. Thank you so much. I think those are all very valuable, lessons and very, very good advice. Now, the hardest question I have for you is, what should we title this episode? I think that's the most difficult one. And I've told this story on the record multiple times, and I'm just going to open my window to capture this. But the story goes as follows: is when we started the podcast, I would come up with record episodes and I would come up with titles based on, the lasting impression of my conversation with the guest. And after a while, it was just too much. It was very difficult for me to, come up with a title. And then my team said, no, Yamina, we need to absolutely come up with interesting titles so that people want to listen to the podcast. And I said, great, I'm happy to do it, but let's talk to the guests after we finish our conversation and ask them this last question. And obviously it's a collaboration. We can both, contribute to it and go from there. So
thank you so much. Yeah,
of course.
I think you are probably in a better position because you have, looked at all the podcasts, and maybe you are in a better position to come up with a title that it is more attractive. But just to keep it simple, I would say a fun chat with a GPCR guy.
Oh, I love it. I love it. I think it's great. Yeah, because typically, what I typically also do is I go through what we discussed on the impressions I had about the podcast. And to your point, we just had a fun chat. I love it. I think it's wonderful. I see you're very creative. I thought you was going to say, I don't know what you want me to call it, but wonderful. Wonderful. Thank you so much. It was really nice having a chat with you. I really appreciate your time. Don't go anywhere. I'm going to stop recording in a second.
Thank you so much for having me. It was a great pleasure talking to you.
Thank you.
Thank you to Dr. Sedaghat for this thoughtful, grounded, and genuinely generous conversation. His perspective is a powerful reminder that science moves forward through patience, focus, and the pleasure of finding out new things. If this episode resonated with you, here's how you can support the show. Share it with a colleague or trainee who cares about GPCRs. Subscribe to the Dr. GPCR podcast so you never miss a conversation. And leave us a 5-star review. It helps the show reach scientists who haven't found us yet. The Dr. GPCR University continues to grow alongside this community through shared learning, open contribution, and expert-led courses inside the nonprofit ecosystem. If you want to help shape what comes next, the link is in the show notes. Thank you to our friends at Celtaris Research for supporting accessible live cell GPCR tools, and a warm welcome to Eurofin's DiscoverX. We're excited to have them join the ecosystem and support conversations that connect assays to real discovery decisions. Thank you to the Dr. GPCR production team for making these conversations possible. Because receptors don't work alone, and neither should you. Until next time, stay safe.